participants, welcome to this parallel session on epidemiology and spreading. I'm Georgios and I will be the chairman of this session. If you want to ask some questions, we can do it at the end of the presentations. Please, you can inform me in the chat in order to keep an order. And I will ask you at the end to unmute and ask your question to the speaker. There will be 4 presentations in this season, and we will start with our invited speaker, Celia Anteneodo from the Pontifical Catholic University of Rio de Janeiro, who will speak about spatial organization and survival of biological populations. Dear Celia, please.
Hello. So I will share the screen.
Yes. It's okay.
It's okay?
Yes.
So as you see, I am not going to speak about epidemiology, but so I can start. I can start, George?
Yes, I think.
Okay. So what I'm going to talk about is about some problems in biological population dynamics. I will make a kind of overview of several works where we focus on model that is a single species model, and looking for the impact of noise heterogeneities, both temporal and spatial, in pattern formation and also in the survival of the population. I just want to mention my collaborators, who are former students, and also Mike, who has a postdoc position. And we have also a collaboration with the people, Emilio and Cristobal, the people of the group of population dynamics in Mallorca, Spain. So just to put this overview into a wider context to motivate, just mention the main problems in population dynamics, more related to ecology in this case. The first question is about survival or extinction of the population. Of course, one is interested in knowing the fate of the population, concerning if it will survive or not under certain conditions. One tries to determine the tipping points that will separate the regimes of survival or extinction. And even if the population, one concludes that it's going to go extinct, it's important to know the mean time to extinction. And these questions are valid both at the large scales of ecosystems, but also at the short scales at the lab. For instance, one studies bacterial populations. And the other issue is related to the spatial organization. One looks at certain species, for instance here, plants or termites, or bigger animals, even human beings. One sees some patterns and patterns which are between regular and random. And one tries to understand the shape of these patterns and the origins of the pattern. And these 2 big questions, survival, extinction, and the spatial organization, are not independent. Of course, in some cases, the structure of the environment will determine if the species survives or not. Or in other cases, the species tries to self-organize in a way that will guarantee survival. For instance, there are recent works showing that connecting habitats prevents extinction. So this is the idea that larger habitats will be better. But this is a controversial issue. For instance, here is another showing that actually small habitats may play a crucial role for some species. So this is the wide context, but of course we are trying to solve much easier problems by now. As a concrete example of what I'm telling, I want to show you some results in experiments with the bacterial populations. Here is the case of a kind of container which is long, can be considered almost one-dimensional, where bacteria can grow. These are simple bacteria in the sense that they are not chemotactic, Escherichia coli. And there is here a photodetector that allows to measure the population density along this container. And the bacteria are subject to some kind of harmful effect, in this case, ultraviolet irradiation. But there can be also a mask, like a refuge that can protect the bacteria. So in this experiment, this is the kind of profiles of the population density that are observed. And the first thing one sees here that calls the attention, actually this is not treated in this paper, but was one of the points that motivated us to study this line, is to see this structure. These are, but it seems rather noise, but if you look and make an analysis, Fourier analysis, there is some characteristic wavelength that is reproducible. So why? This is clearly not uniform and non-completely random. Why this kind of structure forms? And what is treated in this article, besides this point, is the size of the mask. Actually, there is a mask that protects the population, but this will not guarantee that the whole population will survive. There must be a minimal critical size for survival. So this simple example motivated some works and illustrated two effects I posed at the beginning in a wider context: survival and habitat structure, and at the same time the formation of structures, patterns. So how to approach this kind of problem? And either we will see not so simple ones, is a way is through what is called the FKPP equation, which is a standard model for population dynamics, where as we see, we have the typical term, logistic or Berghulst term, where there is growth. Here we are considering a positive coefficient to guarantee that population will grow. And there is also competition, intra-species competition term. The individuals of the same species compete for the limited resources. And also this model considers some kind of movement, which is random movement, then it's considered through this diffusion term. So it's a reaction diffusion equation for a single species. And but although simple, it includes the main elementary processes for population dynamics, and it has some solutions, a trivial solution, which is an absorbing state, unstable, and for the positive coefficients, there is a solution which is uniform. Of course, in this case, we don't have patterns. But another thing I want to call your attention, this model, I picked here a picture of a recent talk by Derrida, where this kind of model actually is used in other contexts, not only in population dynamics. So some of the results perhaps can be applied in a wider context beyond what I'm going to focus here. But coming back to the population dynamics and starting from the FKPP equation, these terms are all local, in the sense they are at most in the scale of the diffusion. However, there are processes particular competition, but also it may be growth or even the mobility term, can be non-local in the sense that here we are considering a single species, but there may be other species, other chemical substances, other things that may form a network of mediators. And in this sense, individuals may compete at a distance or interact at a distance more generally. So for instance, to illustrate, in the case of trees, one can think the roots will pick water and nutrients that may be underground and are shared by trees, even if they are not first neighbors, okay? So in this case, I will restrict to the case of the non-local competition, but also it may be in the other terms, as I say. And to take these non-local effects into account in an effective manner, one can substitute this reaction term by some kind of product by a convolution, indicating that individuals interact with within certain area, and this is through a convolution with the what we call influence function that may have different forms according to the particular interaction. So one gets what is called the non-local FKPP equation, where just we have here this the non-locality concentrated in this term. But when doing that, we are introducing a length scale that eventually may give rise to patterns. And in fact, one observes, for instance, in 2 dimensions, the emergence of structures. Here I am illustrating the case of 1 dimension. In general, the results I will show are in 1 dimension for simplicity, and the formula, mathematical formula and derivations are easier, but in general, they are also observed, the same qualitative effects are observed also in 2 dimensions. And here here is the time, here is the spatial length that in principle we consider periodic boundary conditions simulating an infinite habitat at first, and starting from a distribution, a density which is almost uniform unless small fluctuations, we see that structures develop after time. And this can be analyzed, for instance, through linear stability and obtain the mode growth rate, which will indicate if there are modes with can become unstable or not. And this will indicate in which cases we can have patterns or not. For instance, in this case, where I consider square function, this is the form, the Fourier transform appears. And of course, here is a minus is lacking here. The diffusion is always homogenizing. We'll try to destroy patterns, but the shape of the influence function may determine that patterns are formed. The shape is important. And what I want to focus here is not these details, but to call the attention that this simple model with non-locality may allow to describe, for instance, this experiment I have presented to you. One can explain the appearance of a critical size for survival, and also qualitatively the kind of structures that are observed with the maximum of amplitude near the frontier, the interface, for instance. Okay, but as I said, I will show some results concerning the impact of noise, and also some nonlinearities, additional nonlinearities that we introduce in the equation. So one thing is noise, because of course here we were considering a constant growth rate, for instance, but actually there may be fluctuations in time and also in space. there are maybe additive terms representing external sources of fluctuation. So here I presented more general equation. Basically, it's the same I have shown to you before, but here we introduce some fluctuations in the growth rate, and here an additive term. So by doing that, I will skip some mathematical details, but then we can talk, or you can ask. I will focus on the effect. Here we observe a kind of noise-induced transition, and we observe the appearance of coherence, even when in the deterministic case, it's not expected to occur. And here I am showing again this mode growth rate will be a reference to understand what's happening if there are some modes that grow with a positive rate, there will be patterns. Otherwise, if all modes are negative, there will not be pattern. And here the noises are cancelled. And we follow the average density to have an idea if the population is surviving, and the average amplitude to know if there are patterns or not. And I will show first what happens when in the deterministic case, in absence of noise, there are patterns. So here is the case where there are patterns, and I separated to simplify the case where noise is only additive, or the case where noise is only multiplicative. Of course, we analyze the combined cases. And what we see is with the additive, these full symbols is the average amplitude. So one see that when the noise is 0, there is an amplitude, there are patterns, but then it decays. Noise in this case is extractive of the pattern structure, although the average density is kept more or less constant. And in the case of the multiplicative, is even more destructive, because here you see that not only the amplitude, but also this average density after a critical value of the amplitude of noise falls to 0. So the population is killed. Okay, so it's destructive. Oh, but differently, if we look at the case where there are no patterns in the deterministic case, it is as if noise makes emerge this structure that is submerged in some way, and we see that the amplitude of the average amplitude, which is 0 in the absence of noise, increases up to a certain level. The same, of course, with the multiplicative case. So after...
Macht ihr mal Pause gerade, Kinder, ich muss da mal gerade anrufen, ja?
Sorry? Sorry?
Was ist das eigentlich für einer? Der redet immer mit so einer monotonen Stimme. Ja. Ja.
Please.
Aber der redet immer, finde ich, mit so einer total monotonen Stimme.
It's not with me.
Okay.
Okay, I go on. Can you hear me? Okay?
Yes, it's okay.
Okay. Summarizing, what we observe in this system is that fluctuations, we can say, of course, there are details, but in general, what we see is that these kind of fluctuations are constructive, both for survival and also for the issue of patterning. There are other points we studied I want to mention is that the feedbacks in growth and diffusion, when growth and diffusion are in some way density dependent. For instance, in the case of diffusion, well, it's known that species do not diffuse quite completely randomly, but there is some kind of, in general, subdiffusive behavior in some species, maybe super diffusive, and there is also some response to the density in the behavior of the mobility. And a way to take this into account is through this kind of nonlinear diffusion, which corresponds to some diffusion coefficient, which is not constant, but depends on the density. And this allows to reproduce for the exponent one gets a normal diffusion, and one can obtain the phenomenology of super absorbed diffusion. And microscopically, this means some reaction to the density, in the sense that in one case, the case that produces super diffusion, there is some more activity in less dense friction. There is some reactions to sparseness, while in the subdiffusive case, what happens at the more microscopic level is some reaction to overcrowding. So this, the idea is to study the effect on pattern formation and also in survival of this effect, together with also nonlinearities in growth, because when up to now I have shown the typical case, the standard case, which is with the growth rate per capita, which is constant. But actually there are species that receive a feedback to reproduce more or less according to the density. There are parasites, for instance, where the number of eggs is inverse to the population density, and in algae effects and other cases, one sees a growth which is stimulated by high density. So also a simple way to try to simulate these behaviors is through a power law growth density per capita. So we consider these two nonlinearities in growth and also in the diffusion. So we have our space represented by these exponents of the power laws. And let me show first how they affect patterns. I have here a slide summarizing the effects. The patterns obviously will be affected by these nonlinearities, both the region for pattern formation and also the kind of patterns. Particularly, we are interested, I want to call the attention on a kind which is what we call fragmentation. Here I amplified this, where what one observed is the emergence by self-organization of disconnected regions, disconnected patches. Here there are regions which tend to zero density, and this means that there are no contact processes are not possible in that case, even though the population is interacting, because this emerges in the context of the non-locality, which implies some interaction at a distance. So individuals through the mediators are interacting, but although they are not in contact. So I find interesting that this kind of disconnected clumps can emerge by self-organization of the population, but it depends on the feedback mechanisms related to diffusion or growth or both, which have an interplay. Let us go to the problem of habitat size that I mentioned in connection to the experiment with bacteria, with the size of the mask, the refuge, the region where the conditions are viable is controlled. So here we have a viable, non-viable region. For simplicity, we consider the non-viable completely non-viable, so there are no the population is zero outside as a condition. And in if we linearize the equation, which is valid because we are looking what happens for very low densities to see if the population will finish going to extinction or not, and making, for instance, solving the problem through a Fourier expansion, we see that there are modes that can grow, other not. In particular, if L is very large, the negative contribution will be small, so there will be modes that can have a positive growth. While if L is small, this will be large, so one can have a negative growth for almost all nodes. this is a way to understand what is happening. So there is a critical size. If the size is smaller, one will have all modes fading out. And if the size is large, growth will be possible. Of course, this growth will not be limited, then will be limited by the nonlinear terms we're dropping out here. But what happens if there are the kind of nonlinearities I mentioned to you, both in diffusion and in the growth? I will skip details and show directly this diagram, which summarizes what happens in terms of the diffusion. Here, I highlighted the regimes where there is subdiffusion, and the growth indicates that the growth per capita increases in this direction. So there is a region where we observe essentially the same behavior observed in the linear case. It's this red region where a minimal size is necessary for the survival of the population. But there are other regions that may happen. This is because this survival comes from the balance between the diffusion at the boundaries and growth in the bulk of the patch of the habitat. So depending on the feedback mechanism of these processes, there may be survival and what we call unconditioned, independent of the size. But there may be also a region where we observe that, on the contrary, is necessary a small habitat for survival. And this goes in the sense that I presented at the beginning. Of course, we are not pretending to explain this more complex problem, but how not always a larger habitat may be favorable. I don't mean that the mechanisms are the same, but we are presented some processes that can explain why, dependent on the feedback mechanism, it may be more favorable to be isolated or to be sparse or concentrated in a small habitat than sparse in a larger one. And since, of course, since there are many nonlinearities, the dependence on the initial conditions will be strong, which will indicate details that I will not mention now, but that may be important at the time of, for instance, reproduction species for repopulation. I also want to mention rapidly that through this kind of model, we can also tackle other problems like the so-called area diversity curve. In many cases, it's observed that in general, the number of species increases with the area. Of course, here we are dealing with a model that is single species, but we can consider a set of equations where we also take into account the interactions between the species, and in this way we can reproduce the kind of laws that are observed in the so-called area diversity curve. Another thing I want to mention is a recent result where we studied heterogeneous diffusion. Previously, I have shown the effect of the state-dependent diffusion that depends on the density, but one can be also interested in knowing the effect of a diffusion that depends on the landscape position. And so we have an amplitude that kind of nonlinear multiplicative noise that depends on the position. And we constructed the corresponding diffusion reaction equation for this problem. And we have shown mathematically that independently of the distribution, if there are heterogeneities in the diffusion coefficient, we observe in a general way that a patch size reduction for survival. The minimal critical size is reduced when there is heterogeneity in diffusion. And there are some details. For instance, here we have assumed a Stratonovich kind of prescription for this multiplicative noise, which is more suitable for these kind of problems where there should be some kind of minimal correlation or anticipatory noise. But we see, for instance, if we have Iton noise, the effect is contrary. A noise that is completely uncorrelated will produce the contrary effect, and we see that the critical size with respect to the one of the homogeneous case increases. A larger size of the habitat is required in that case. I have time for, I want to show perhaps the last result, which has to do with heterogeneous environment. And I have shown what happens for the critical size, but now I want to show what happens related to pattern formation. Of course, we have a critical relation for the survival of the population, but under certain conditions, patterns can emerge, and these are the kind of patterns I have shown at the beginning related to the population of bacteria profile, having a larger amplitude near the interface. And it's interesting that we put this interface and we analyze the growth mode, even when all modes have negative growth rate, then we will not have patterns in a homogeneous environment. Here we have difference in the growth rate in particular, and we observe a spatial profile where patterns form, while in the homogeneous case, they are not formed. So there is also in this case a constructive role of the heterogeneities, in this case, heterogeneity of the growth rate along a space, where these kind of patterns develop by the presence of the interface. This is also observed in 2 dimensions, but as I said, I am restricted to some analytical results in 1 dimension, where we can describe these profiles analytically. And especially, I want to focus on the following. So here you see, of course, for certain values of the parameters, we have, when in the homogeneous case, here all is viable, we have patterns or not. But when we have an interface, a discontinuity, we observe that patterns can develop, but they may be sustained or not. They may decay with certain characteristic distance. Just how much time? Negative?
You have 1 minute about.
Okay, let me skip this. Analytically, we can make a mapping between the shape of the interaction function, the function for the which represents the interaction, and the kind of patterns observed. This is analytical, and reciprocally. If we observe in nature patterns with certain characteristics, this allows us to come back and see, infer information about the interactions, to see if this is robust or depends on the kind of kernel proposed. We have done this for kernels with different shapes, and what one observes is that there are some characteristics, of course, the full details, we will need to have the full spectrum to infer, but certain characteristics like compactness, characteristic length of the interaction are rescued by this process. So I will skip some results about metapopulation and pass to this summary. So we have studied certain generalization of this single species model. introducing nonlinearities, spatial and temporal noise, heterogeneities, and looking for the tipping points that will lead either to pattern formation or not, to survival or not. And as general conclusion, we can see that in this kind of model, at least noise heterogeneities are constructive, may make appear patterns when they will be absent in the noiseless or homogeneous case. So an interface, for instance, one can see as a kind of lens that if one approaches or looks near an interface may see information which will be hidden otherwise in the absence of heterogeneities. Well, this is more or less what I try to transmit to you.
Thank you very much for this nice talk. We have time for one question, maybe. If you want to ask a question, please unmute and do it. No, I think there is no question. Just I want to ask about the noise you added in your equations. Although I'm not familiar with these studies because generally I work with dynamical systems, deterministic dynamical systems. I can't understand what is the physical origin of this noise and how you distinguish between additive. You say you have additive or multiplicative.
Well, yes, this will depend on, let me show. We have parameters in the basic standard equation. We have constant parameters, but it's reasonable to think that in a biological system have a complex landscape, these parameters will not be uniform and will not be constant in time. So it's very reasonable to introduce fluctuations around the mean values. So this is the main spirit of this, to take into account in an effective manner the heterogeneity of a complex landscape and the variations in time too, okay? So, and dependent on the parameter we fluctuate, this will lead to a multiplicative or additive noise as a consequence, okay?
Thank you very much. So we continue with the second talk, which is by Iacopo Pozzana from the University of London.
Yes.
So,
hello.
Iacopo, please share your screen and...
Gladly. Now, I have not had as many opportunities to do screen sharing on Zoom during these months, so you will forgive me if I am a bit clumsy, but can you see my presentation now?
It's okay, it's okay. Yes.
Perfect. So let me thank the organizers and let me thank Celia for a very interesting talk. I'm going to actually follow in her footsteps, and I'm also not going to talk about epidemics. Indeed, I'm going to talk about project management. My name is Iacopo. I am a PhD student at the University of London, but I am also a data scientist in a startup called Nodes and Links, where we apply complexity science and network science to the field of project management. So why is this relevant? Well, it's a huge industry that 6% of the entire global GDP is spent on large projects. When I talk about project management, indeed, I'm mostly referring to large construction project or infrastructures, and this is because this industry is highly inefficient. So on this 6% of global GDP, roughly a third, 28%, goes lost in overruns because of headlines like this one that are probably familiar to you. A pretty commonplace. I don't suppose that any one of you is shocked by learning that some huge infrastructure is delayed by 5 years or by several billions. So this is a field, is a huge industry, and is badly in need of rescue. So what can we do about it? As in, what's the role of networks in this respect? Well, in a sense, a project can be seen as a series of activities that need to be carried out. I have a very simple example here. If you need to send some invitations for your birthday or whatever, you can break it down in smaller activities. You need to design the invitation, print them, and address them, and so forth. And these activities are connected, as in that some activities rely on others to be completed before. For example, you need to design your invitation first, and then you can print them. And so you see that we have a natural network structure where your nodes are activities, and those activities are connected by dependencies. It's a directed network, and it is an acyclic graph. So it is a DAG, because if you had a cycle, it would mean that you would not be able to carry out your project. So let me just quickly mention that there are other aspects that sort of contribute to complexify the whole problem of project management. You have resource allocation, which can be seen as a bipartite network problem, supply chain, which is another network in its own right. You have costs, because of course not all delays are equal. But we're going to talk about project network in the sense of activity networks today. So I have a little bit of a bibliography here in case anyone might be interested. Which is, well, just to say that there's been some interest in the network community into this application to project management. But what is in particular the problem with spreading? What is it that spreads on these projects? Well, we've been talking about overrun, we've been talking about delays, and so projects, these activities usually come with a scheduled start and an end. So we can measure whether they are delivered early or on time or late. And we can call this the performance of an activity. And obviously this performance propagates downstream along those edges, because if you are late in designing your invitation, then you're going to necessarily be late in printing them as well, no? So this problem can be analyzed as a spreading problem. Also, we are gonna try and provide some sort of form of well, something that can help with a localized intervention, because large-scale interventions are not really actionable in this problem in practice in project management. Because if you cannot just say, well, spend 10% more on everything, because that money simply is not provided. So you need to be able to identify where the problem will emerge, ideally before they emerge. So we're going to try and work on this local level, but to do so, we're actually going to start from the global level. So just to contradict what I just said, we're first going to introduce this. We have introduced this novel definition, but it's actually just an extension, a slight manipulation of an existing measure that was introduced by Strada in 2010 to quantify the heterogeneity of network. But we specialize this to DAGs because we work with DAGs. So this allows us to introduce a more comprehensive description of the network, because we take the sum on all connected pairs. And basically we take the difference between the number of descendants of the node upstream and the number of ancestors of the node downstream, and we weight the difference and we sum it for all possible pairs. Why is this? What's the relevance of heterogeneity? Well, as you know, heterogeneity is bad news when it comes to spreading. A highly heterogeneous network is a very vulnerable network. So this score tells us something about the overall vulnerability of the network. The higher the RH, the worse news it is for the network as a whole. But to specialize locally this global definition, first of all, what do we mean locally? Well, we could be talking about edges, but there is not really much that we can do about edges, because you need to design your invitations before you can print them. There is nothing you can do about it. On the other hand, at the node level, you can sort of provide a kind of immunization in the sense of adding more resources to make sure that the task is carried out, that the activity is carried out on time, or you can improve your surveillance. Also, we have a way of measuring performance at the node level that I will discuss in a moment. So to localize this notion of RH score, we simply remove the node from the network, we recalculate the RH score, and we take the difference. If the RH is high for a node, it means that it adds a lot of vulnerability to the network as a whole. So we have this local measure. How do we measure performance? Let me go with this first because it probably makes more sense. So we have, I mentioned that every activity has a start and it has an end, but also start and end come into sort of different packages. One is the target date and also sometimes the time, depending on the resolution that you're using. And target is when you had originally planned to start or end that activity, whereas the actual start and end are instead the moment when the activity actually started or ended. So by taking this difference, you can measure the performance on an individual activity. The higher the performance, the earlier you have managed to complete. Negative performance means that you're late. Positive performance means that you're early, and so on and so forth. So we're working with we run some experiments on some data sets kindly provided by some of our clients or partners for data sets of rather different infrastructures, difference in size, different in structure. We can talk about this more. I have some, I don't think I have the time to give a detailed profile, but let me jump to the juicy part. We can discuss this later if you're interested. So basically, as you can imagine, we have compared RH local score at the node level versus the performance of the nodes, and a pretty clear pattern emerges where lower RH score corresponds indeed to the best performance of the nodes, as we would have expected. Also, if you look at some of these graphs, in particular the two on the right, you will see that this pattern is driven by outliers, because the center of the distribution pretty much remains in the same place, where it is the mean that is really, the trend is actually apparent in the mean. And so we see that this distribution is actually driven by outliers that are the low performing, the best performing nodes that are low in the RH score. And now to add a sort of further layer of validation to the effectiveness of this score in providing information regarding the performance of nodes. So we have run a sort of comparison, well, a comparison. We have used the mutual information, measuring it between local RH or 7 other candidates, which are node metric levels, the mutual information between those metrics and the start performance on the other side. And we see that the local RH scores the highest in mutual information on 3 of our 4 data sets. And so this is pretty much it. I'm glad I managed to save a couple of minutes. So basically, just to sum it up, we have introduced this measure. The RH is a topological measure specialized for DAGs. It's content agnostic. In principle, could be used for other type of epidemic processes. But we have found that it's actually quite informative, highly informative when it comes to the performance of activities on project networks. In particular, there is a trend where lower H corresponds to worse performance, but this trend is driven by the outliers, which are the best performing nodes with a low RH score. Let me spend just a quick word to acknowledge the rest of my team, Christos, Georgios, and Kostas, where the data science team at Nodes and Links. And if I can introduce a shameless plug, we've just won a Horizon 2020 grant with collaboration with University of Cyprus. Therefore, we are now hiring for data scientists, and we're also in general always interested in opening new research collaboration. We have a quite interesting wealth of data of networks with this ground truth of sorts for spreading that I have discussed. And that's it. Thank you very much.
Thank you, Iacopo. Is there any question?
Yeah, I had one.
Yes, please.
I just wanted to know if this this methodology or these findings have been applied to clients in project management, like the results that you have been tested on newer data.
We have not yet, we have not provided these results to our clients so that they have not act on these insights yet. This is one of the things that we're going to do in the next month, actually introduce in our product this RH measure. And then obviously we cannot sort of run a proper experiment in that sense, because we cannot tell our client to do this. They decide what to do with the insight that we provide. But we're going to introduce this as part of our product, and we're going to see what the feedback is.
Right. Thank you very much.
Thank you for your question.
Okay, thank you, Iacopo.
Thank you.
We can proceed with the next talk, which is by Sebastian Benthall from the New York University, and with the title Privacy and Contact Tracing.
Hello. I believe you can see my screen, yeah?
Yes, everything is okay.
Everything? Oh,
Yeah.
let me try to do just the window. That should be better. Okay. Thank you very much for having me and for attending. This is joint work with Erez, Hatna, Joshua Epstein, and Katherine Strandburg, and it's a collaboration between the School of Law and the Agent-Based Modeling Lab at New York University. And the motivation for the work is COVID-19. I guess I will buck the trend and actually do a talk about epidemiology. But we've noticed that COVID-19 is the first pandemic to occur when there is widespread use of smartphones. Smartphones were really introduced in 2008 and now are very widespread. And for that reason, there's been a lot of innovation this year in the use of smartphones for contact tracing applications, especially using Bluetooth connections to trace the proximity of different smartphones. Although there's been geolocation-based contact tracing methods as well. And then there's a parallel conversation that's about the privacy concerns about these applications. And people have been proposing new kinds of encrypted contact tracing solutions. As far as we know, we're the first to investigate the interaction between these privacy concerns with smartphone contact tracing and the effectiveness of the contact tracing from an epidemiological perspective. So how much does privacy interact with the public health? A couple of us on the team are primarily privacy scholars. This is my first time presenting at a complex systems conference. So thank you very much for being an audience here. But when we're coming from the context of privacy scholarship, it becomes very important to note that privacy has many meanings based on context. There's no single privacy concern. We've grouped, for simplicity, these concerns into two categories. First, a general vertical concern about the misuse of data by harvesting institutions. So, for example, the concern that the company that operates the smartphone, or that built the app, or the government that did the contact tracing system might do something inappropriate with the data. A different privacy concern is a local or horizontal concern that somebody in one social context might learn something that they were not meant to from another social context. So an example would be an employee of a firm who does a job interview with a different firm while they take a vacation day from their employer. That's a kind of contact that they would prefer to keep private, and which, if it influenced their decisions, might cause them to disable or uninstall a contact tracing system if they believe that this might be the source of a leak. in order to model the connection between these privacy concerns and epidemiological effectiveness, we developed a network-based SEIR model, a compartmental epidemiological model with the 4 normal compartments of susceptible, exposed, infectious, and recovered, but with some additional categories. Agents might be symptomatic. There's a chance zeta of an infectious agent becoming symptomatic. An agent might be quarantined, and they could be an adopter of the contact tracing system. And in each time period, the edge weight is the probability that an edge activates. And when an edge activates, then an infectious agent might infect a susceptible at the other end of the edge, provided neither are quarantined. And that contact, whether or not there's an infection, is traced with a probability of C if both end nodes are adopters. So the adoption rate creates a larger field of tracing. And lastly, if an infectious agent becomes symptomatic, they are tested and quarantined. And a positive tested agent, any positive tested agent, alerts previous contacts to be tested. So at the point where any infectious agent becomes symptomatic, in this model, they become quarantined and trigger a cascade of tests through their traced contacts. We have tested this model on 2 closely related network topologies. This is all theoretical work, and we're just trying to get some of the basic science down at this point. We're using a Watts-Strogatz model, which is, of course, a circular lattice with a rewiring rate for edges that go across the circle, so to speak. And then a closely related 2-dimensional lattice with rewiring. You can think of it as a 2-dimensional version of the Watts-Strogatz model. And we're distinguishing between the links in the original lattice structure and the rewired edges, because we think that's the way to operationalize these 2 different kinds of privacy concern. So the general concern, distrust in the contact tracing system, we believe that will translate to lower contact tracing adoption and or reduced effectiveness of the edges being traced in general. Whereas for the local or horizontal concern, we're interested in the selective disabling of specifically the distant or rewired edges. The idea here being that in one's local neighborhood, in the original lattice structure, your edges are mostly known to your company, whereas a distant edge, a rewired edge, is a higher information edge with respect to your local contacts, and therefore might be more sensitive due to these local privacy concerns. Obviously, this is highly schematic. However, it has led to some interesting results. So the first piece that we rediscovered, there's prior work that's done similar results, is that rewiring rates really matter for the spread of the disease. If the rewiring rate is 1, meaning all the edges get rewired, we're effectively using a random network. And it's much easier for the disease to spread. Whereas if it's strictly a lattice structure, the disease will spread geometrically rather than exponentially to start with. So we know that having distant edges is correlated with a higher rate of infection, or the likelihood of a pandemic as opposed to an early extinction. But what we found is that contact tracing on these distant edges is not particularly effective. So while there's always a benefit to having more traced edges within the contact tracing system, it doesn't seem to matter very much whether those traced edges are distant or close edges, which is a bit counterintuitive, given that you'd expect that if you remove these edges from the network or you made them local, the disease would not spread as much. So for some reason, the contact tracing system is lagging the infectiousness of the disease itself. This is another way of looking at the same data. you know, even if you're comparing, for example, 600 close edges traced and 600 distant edges traced in the fourth panel here, you see there's virtually no difference to whether or not additional edges being added to the system are close or distant. And we think that most of the other differences here are just noise. And so what that means is that the general concern is very problematic for the effectiveness of the contact tracing system. If people don't trust the system and therefore don't install it, then of course they will be less effective at reducing the infectiousness ratio. But the local concern does not seem to be particularly important to the efficacy of the system. If someone decides to turn off the app temporarily in order to do something which is more sensitive with respect to their privacy, that's just an edge that's missing. It's not especially effective. It's certainly worth the trade-off for them installing the application in the first place. So from the Watt-Strogat study now, we found that distant edges are significant for the disease spread, but not particularly for the contact tracing. And so if we should try to address the privacy concerns that reduce adoption generally, and that means the design of the tracing system should allow people to selectively turn off the app for local discretion, because that will improve adoption overall. It's important that people have trust in these contact tracing systems. However, this is all very preliminary. We haven't published the paper yet, and the next step is to test these results on the 2D lattice structure to ensure their robustness. It's all work in progress, and that's the end of the presentation. So I welcome any questions and also especially any feedback of how we might improve this study. Thank you.
Thank you, Sebastian. Are there any questions? Okay, you may ask something to the chat to Sebastian, so we can proceed to the next talk, which is by, okay, Sebastian, which is by Leo Torres. Leo is here.
Yes, hi, hello.
Okay, Leo from the University of Boston. And he will talk about node immunization with non-backtracking eigenvalues and X-Centrality.
Yes, I'm preparing the audio and everything. So let me check that everything's all right. Okay, cool. So please stop me if you know, if anything is wrong. I have a pre-recorded talk and I'm going to start it now.
Yeah, yes. Okay.
Hi, I'm Leo Torres. I'm a PhD candidate currently on my 5th year at the Network Science Institute. Today I'm going to show you two things. First, what happens to the leading eigenvalue of the non-backtracking matrix when a node is removed from the graph? And second, how to use this knowledge to define a notion of node centrality and use it for the application of node immunization. But before all that, let me convince you that you should at all care about this matrix and its eigenvalues. Say you have the graph on the left with m edges. Then its non-backtracking matrix B will have 2m rows and 2m columns, so 2 rows and 2 columns per edge. This matrix is a transition matrix of a random walker that does not trace backtracks. That is, it never traverses the same edge twice in succession. For example, going from the red node to the blue node and back to the red node is a backtrack, and that is exactly what we're trying to avoid. Say there is a random walker that does not trace backtracks that just moved from the red node to the blue node. Then the matrix will have a row for the directed edge from red to blue and a column for the directed edge from blue back to red. The corresponding entry will be 0, meaning that the walker cannot go back to where it came from, whereas other entries will be 1 for any other valid step. The eigenvalues of these matrices have multiple applications. For example, they have been discussed in terms of length spectrum theory, community detection, graph distance, graph embedding. They have already been used in the past to define node centrality. But let me focus on this last application. it is known that 1 over the leading eigenvalue lambda is a good approximation of the epidemic threshold of certain kinds of spreading dynamics, like the SIR model, for example. Remember that the higher the threshold, the more difficult it is for a disease to become an epidemic. So we would like to make the threshold as high as possible, and thus the eigenvalue as small as possible. Which leads us directly to the main question that we're dealing with here. What happens to the eigenvalue, and thus to the epidemic threshold, when a node is removed from the graph? In general, the eigenvalue will decrease, but we want to quantify by how much so that we can choose which node to remove if we're in a node removal or node immunization kind of setting. So let's get right into it and see how we can come up with an answer. First, let me write down some notation. In this graph, I will try to remove the node labeled as C, which I will call the target node, and I will color all of the edges that are not incident to C in blue, while all of the edges that are incident to C are in yellow. After removing the target node, I get the graph on the right. Now, we're going to use symbols with the superscript C to refer to the graph before the removal, while every symbol without the superscript corresponds to the graph after the removal of the target node. Essentially, what we're asking here is what is the difference between lambda 1 C and lambda 1? Let's start with the graph before the removal, and let's arrange the rows and columns of its non-backtracking matrix so that the blue edges, those that are not incident to the target node, form a block at the top left, and the yellow edges form a block at the bottom right. We're going to label the blocks as B, D, E, and F, and note that the blue block B at the top left is exactly the non-backtracking matrix of the graph after the removal, since by definition it depends only on the edges that are left intact. With these blocks, we define one more matrix X as D times F times E. This matrix turns out to also be a binary matrix that indicates the existence of non-backtracking paths of length 4 of the type blue, yellow, yellow, blue. These are exactly those paths that are destroyed when the target node is removed. So in the top left block B, we have the structural information that is kept after the removal, while X contains the structural information that is destroyed. Using this setup, we can now focus on trying to solve this equation. By definition, the solutions to this equation are the eigenvalues of the matrix BC. I will give a quick overview of the steps involved, so for more details, please see our paper. First, I'm going to apply a well-known formula for the determinant of a block matrix, which after some massaging and some improving some lemmas, will look like this. Now in this formula, I'm going to focus on the third term on the far right here, which depends on the matrix Y, which is defined in the bottom left of my slide, and also on the matrix X, which is defined in the previous slide. The point here is that if I make this third term equal to 0, then the whole thing equals 0, and I will have found one of my eigenvalues. So this is what I'm trying to solve now. And what I'm going to do is linearize this equation and neglect all of the small terms. The leading term here involves the trace of y times x. But the trace is nothing more than the sum of the diagonal entries, which can be written as this. These terms now involve ui and vi, which are the right and left eigenvectors corresponding to lambda I. Now from these terms, I will take only the first one, since all others are small as well, and I'm going to forget about all of those small terms and focus on the terms in the orange box. I'm going to set all of this equal to 0 and rearrange to finally arrive at this expression. And now I'm done, because this is a third-degree polynomial on the variable t, which can be solved using a closed formula. However, we can do even better, because the value of the constant term will determine the solution. If this constant term is large, then the solution is large, and if it is small, then the solution is small. And that means that in order to estimate what happens to the eigenvalue after removing the target node, we do not really need to actually estimate the eigenvalue, we just need to estimate this term. That's why we define this term as the X non-backtracking centrality, or XNB of the target node. In the paper, you will find another procedure that establishes that the XNB centrality is upper bounded as shown here, where the terms in the 3 dots can also be neglected. Here we focus on this term, which is not only an upper bound, but also happens to be correlated to the x and b centrality, and so we call it the x degree centrality. By the way, all our derivations are valid on any graph without restriction on their structure. In particular, we are not making the assumption of absence of short cycles, or the graph being locally tree-like that is frequent in other derivations. So let me real quick go back to our main question. What happens to the eigenvalue when you remove a node? It decreases by a quantity that is correlated to the x and b centrality. How correlated? Very correlated. Here in the vertical axis, you have the true change in the eigenvalue after removing a node, and on the horizontal axis, you have the x and b centrality of that node. Each marker on the plot is a randomly chosen node from a randomly generated graph in the stochastic block model. And as you can see, there is a strong correlation, and the same pattern is repeated for other kinds of graphs. Yes, the degree is also related to the change in the eigenvalue, but the correlation between x and b and the eigenvalue is much stronger, in some cases going up all the way to almost 1. So let's talk a little bit more about these centrality measures and see how they can be used for node immunization. First, let's talk about how to compute them efficiently. Up to now, everything we've done depends on having fixed the target node. So if you fix the target node and rearrange the matrix, you can define the X matrix and then compute the centralities and so on. Here, by the way, I'm using the subscript C in the X matrix to highlight the fact that if you choose a different target node, the matrix X will be different. With this matrix and the eigenvectors, you can compute the X and B like this. But if you rewrite this equation in scalar form, you will find this formula where the A's now come from the adjacency matrix and the V's are the standard non-backtracking centralities of each node, which is another centrality measure that was defined years ago. So to compute the X and B of all nodes at the same time, you do not really need to rearrange the matrix each time and then compute a different X matrix each time. You can just compute the vector of standard non-backtracking centralities of all nodes and re-aggregate them using this equation. The same can be said for X degree. This is the original definition, and this is a scalar form, where now these coefficients d prime are nothing more than the degree of the node minus 1, also called the excess degree. This is why our centralities are called the X non-backtracking and the X degree centralities, because they start from these more standard centrality measures that are then aggregated using the X matrix. Let's talk about the application to node immunization. This is a procedure where you are removing nodes one by one according to their centrality in order to decrease the leading eigenvalue as much as possible. The table that I'm going to show you presents the percentage decrease of the eigenvalue, so larger is better, averaged over repetitions in different kinds of synthetic random graphs after removing 1, 2, or 3% of their nodes using many different centrality measures. Here, NS is the net shield algorithm, and CI is the collective influence centrality, which is yet another centrality measure also based on the non-backtracking matrix. These are the results where you can see that degree is actually a very strong baseline for this task, and that our X centralities are in general slightly better than the others, especially in these kind of random graphs, though the difference is statistically significant in all cases. So I just want to finish by pointing out a few interesting properties that we think are good lines of future research. Let's take this scalar formula of X degree as an example. Like I said, this depends on my neighbor's excess degrees. But now let's compare this to minus the variance of my neighbor's excess degrees. These are almost the same formula except for the normalization terms in the orange boxes. Now, compare this also with the collective influence centrality that I was using in my experiments before. The CI is also defined in terms of my neighbor's excess degrees, but now it is proportional to a linear combination of them, so it is a function of the first moment. So my question here is, what happens with the higher moments of my neighbor's excess degree distribution? Perhaps there is something that both CI and X degree are missing, or perhaps there is another matrix somewhere out there that will care about the third or the fourth moment of my neighbor's degree distribution. So to recap real quick, the leading eigenvalue decreases by an amount that is correlated to the X and B centrality when you remove a node from the graph. We have developed here new techniques to analyze the non-backtracking matrix, using the X centrality to remove nodes in order to decrease the eigenvalue is slightly better in general, but substantially better in some cases. I want to acknowledge and thank my collaborators, Kevin Chan from the Army Research Lab, Hanghang Tong from the University of Illinois at Urbana-Champaign, and my advisor, Tina Eliassi-Rad. Thanks.
Thank you, Leo, for your contribution. Are some questions for Leo? So, okay, I think we can close here our session. I wish you have a nice evening or a nice day for people, at least. Bye.